In On the Origin of Species, published in 1859, Darwin succeeded in clearly presenting the mechanism of evolution and was the first person to convincingly explain where species come from and how they change. However, the theory of natural selection, which would shake the world and fundamentally change the history of science, had a fundamental problem. He had not been able to solve the mechanism that made intergenerational information transfer possible, allowing evolutionary changes to occur. Well, perhaps no one knew or even came close to knowing this mechanism, except for a quiet Austrian monk biologist named Gregor Mendel. Like the vast majority of his contemporaries, Darwin was completely unaware of Mendel's genetic studies. Therefore, he had to make his own guess. He theorized that particles called gemmules circulating in our body collected samples from every point of our body and then carried these samples to our sperm and eggs, allowing the parents' information to be passed on to the offspring. As you know, what actually happens is quite far from this. 10 years after Darwin's masterpiece, in 1869, Swiss biologist Friedrich Miescher, while working on white blood cells, succeeded in isolating a molecular soup that included DNA. Of course, it was not possible for him to know that a molecule called DNA existed at that time, but even being able to isolate a mixture containing this molecule was a great victory. Because today we know that DNA, or by its full name deoxyribonucleic acid, is an extraordinary molecule found in almost every cell of ours, controlling the development, function, and reproduction of the organism, and carrying nearly all the information about that organism. You can think of it as an encyclopedia carrying the necessary information to build a living being, but perhaps more importantly, DNA is the molecule that establishes the bridge between parent and offspring, ancestor and descendant, thus making biological evolution possible. Here, Miescher had named the mixture containing DNA as nuclein and had accurately thought that this soup might be related to heredity. Unfortunately, Miescher's discovery and his name were lost in the dusty pages of history, and his success went unnoticed by anyone for many years. Fortunately, the 20th century became the era when such peculiar geniuses were rediscovered. First, in 1900, Mendel's works resurfaced, and the field of science we know today as genetics was born. In the following years, studies first illuminated the foundations of genetics, and then revealed the relationship between evolution and genetics, resulting in the remarkable scientific revolution we know today as the modern synthesis. Thus, due to these developments and the rediscovery of Miescher's work, by the year 1953, which marks the starting point of Watson's great fame, biologists largely knew that DNA was at the center of genetic inheritance. The problem was that they had not been able to figure out how DNA encoded all this information about the organism, how this information was transmitted from generation to generation, and how it controlled the activities of genes within the cells. In order for all this to start being resolved, first, the physical structure of this molecule called DNA, that is exactly what it looks like, needed to be determined. Until that day, no one had been able to achieve this. And this meant a tremendous scientific opportunity. Therefore, Watson, who had just arrived at the Cavendish laboratory, had only one thing in mind: to illuminate the secrets of DNA, which he was convinced was the most important topic in biology. Meeting Crick at Cavendish, who had the same goal, would provide him with a great companion.